Hi everyone, this is Jason, producer of Data and Biotech. Before we get started, I wanted to let you know about our latest white paper. It's a comprehensive guide to implementing machine learning models in biotech manufacturing. It's a complete overview of all the potential problems of ML adoption and, more importantly, how to solve them. To download it, simply visit connect.cordine.com/biotech-ml. We've also dropped the link in the show notes of this episode. Okay, let's get into it. Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. In this episode, we talk with Amy Gamber, Vice President of Manufacturing at Atara Biotherapeutics. Amy shared her insights into the complexities of manufacturing cell therapies, highlighting the importance of managing donor variability, scalability from trials to commercial production, and continuous process monitoring. She and Ross also discussed the necessity of automation to facilitate advanced data collection and the potential of AI in biotech manufacturing. Here we go.
Amy Gamber, welcome to the Data and Biotech podcast.
Thank you very much for inviting me. I'm very happy to be here.
Awesome. Well, just to kick us off, could you give us a brief introduction to your background and what brought you here today?
Sure. So, as you said, I am Amy Gamber. I am the Vice President of Manufacturing at Atara Biotherapeutics. And in this role, I manage a network of contract manufacturing sites for both clinical and commercial cell therapy programs. And that function includes external manufacturing as well as process engineering and manufacturing support.
Awesome. So can you tell us a little bit about Atara Biotherapeutics and, the work you do and how that relates to the manufacturing processes you're talking about?
Absolutely. So Atara is an allogeneic T-cell company, and our mission is kind of twofold. So the first is we have our allogeneic cell therapy product, which is tabelecleucel or tab-cel. That's kind of easier to say. And it's the first allogeneic T-cell therapy approved in the world, which is really exciting to be part of that. It has a really great efficacy and safety profile. We've treated 500 patients. And the thing that's really, for me, speaks about this is that without this treatment, patients may die in 30 to 45 days. It's a particularly cruel disease for the indication. And so we've got a therapy with this that can be dosed within 3 days at a commercial GMP scale, which for a cell therapy, that's great. And it's again, because it's kind of off the shelf, mission. And so with that, then we're also then leveraging that tab cell experience to build on it with our early stage portfolio with CAR T programs to really focus on unmet need in the oncology and autoimmunity spaces. We have 2 INDs for our CAR T programs, and it's again allogeneic CAR T, and we're continuing to develop our pipeline in this area. And for me, that's the challenge, is managing both a commercial scale in 2 jurisdictions eventually, both the EU and the US, as well as small scale early phase 1 programs.
Interesting. And allogeneic in this case refers to donor derived. Am I right?
Absolutely. Yeah. So allogeneic is you taking the cells, your donor derived from healthy patients, whereas autologous, which is also another arm of cell therapy, is you're taking the patient's own cells and then using them for treatment.
Can you just walk us through the process for how you go from the very beginning of getting the donor cells all the way through sort of the manufacturing process and the delivery to the 500 patients that you're talking about? I think that might help to frame sort of the manufacturing conversation later on.
It's actually interesting because it's not a sequential process. So we take the donor cells, or we take the leukapheresis, and we call them leukopaks just for ease. And we take that from a healthy donor and we convert that into a material that's called a PBMC, a peripheral blood mononuclear cells. And that is one of our starting materials. And so that's kind of the basis for our program. From there, we convert it into another intermediate. And both of those, what's kind of unique about allogeneic is both of those starting materials are then frozen and stored. So they're cryogenically preserved and they're put in storage. And then when we're ready to run a manufacturing campaign, I can pull those items out of storage and then I run them through about a 40-ish day process. But again, I can run that ahead of time on my own schedule, much like you would look for like a biotech or even a small molecule campaign. And I can then cryopreserve that and have it available in inventory. And then when a patient then presents needing the treatment, we go through a matching process. and we're able to then pull, ship, and treat the patient within roughly 3 days. So that's a huge improvement, where some of the autologous programs, you have to actually take the patient's cells and go through the process of making the drug and then give it back to them, which sometimes can take a lot longer.
Right. So it sounds like there are points in the process where you can sort of like store in inventory so that you can, like manufacture on your own timeline and manage your inventory accordingly versus having to sort of like on the fly manufacture and ship directly to the patient. Am I thinking about that right?
You've got it exactly right. We're able to go in and pick our manufacturing slots. We can look at our inventory, we can look at coverage, we can look at patient, projections, and then we can decide, okay, I need to run a campaign of, 6, 12, 20, whatever lots. I can schedule that ahead of time and have that done and stored and ready to go, well in advance of when a patient's needing the treatment.
Interesting. Can you just talk about some of the biggest challenges that you face in managing a manufacturing process like this?
Yeah, so there's a couple of things. The biggest one is donor variability. So, you're looking at, and I think this is across cell therapies in general, whether you're using the autologous product where you're using the patient's own cells, or you're doing allogeneic where you have healthy donors. In either case, your starting material is in fact a donor cells, and there's so much inherent variability in people, you automatically are introducing variability into your process. So you have to figure out how to control for that. And I think, with autologous products in particular, not only do you have the challenge of having all these different diverse people, but then you're adding in age, immune status, where are they in their disease state, and their immune system can already be compromised because of the illness or because of the treatments that they're going through, like chemotherapy. And that really can add to the variability of the T cell population that can be harvested for further development. And even with allogeneic, you don't have the challenges that you might have with sick patients, but you still have this wide, diverse population. And how are you making sure that, one, you're collecting to get, the donor types that you need to cover your ultimate patient population, but still accounting for that variability. And then the other challenge that we really see is scale. So oftentimes a lot of cell therapy, and particularly some of the products that we're in, at least initially, are orphan drug, which means that they're treating, these these smaller populations that are sometimes in the hundreds instead of thousands or tens of thousands. And so how do you look at, I'm in a phase 1 trial, early phase, where I have a small handful of products. I don't want to make a whole lot of investment into my programs until I know if I have something that works. But then at some point, I've got to translate that into commercial scale, where I need a robust, commercial process that can be inspected and approved by the agencies, and then has to be able to treat, whatever my patient population is at that point. So how do I make that investment? Where do I make that investment?
So you talked about two main challenges. You talked about donor variability and you talked about scale. If it's okay with you, I'd like to sort of take them one at a time. So on the donor variability side, how do you think about, I'm assuming, segmenting the population or like parsing out the variability in a way that allows you to gain control over that variability and understand, which donor samples to match with which patients?
So there's a couple of things. One of them is really what we've learned through our tab cell experience is how to better manage the variability of our donors. And the way we do that is we've learned that you can define those critical quality attributes in your PBMC process. And from that, we know how we can start to pre-screen. So we know what we're looking for, and we now do a small scale production process. And we can look at things like alloreactivity. We can look at what is the capability of those cells of becoming an EBV CAR T. Can it be transduced? And we're now applying those lessons to our early phase CAR T. So that's one way where we're starting to be able to control that donor variability. The other thing that we look at is we look at those patient populations, and we're looking at what are the different, HLA types that we need. HLAs are kind of the markers, if you will, within your, genetics or your cells. And that tells us, okay, we need more of this particular HLA type because we have 20 or 30% of the population potentially would match to that. So then we can go to the donor sites where we're using for the collections and say, this is the profile that I'm looking for. Can you help us find donors that can match that particular population? and then we bring them in for collection. And again, we're doing this because we can do it ahead of time. We can look at and we can forecast, well, we think in 2025, 2026, we're going to need XYZ patient populations to be treated. So we can do this all well in advance of when we would need the actual product.
Yeah, this is interesting. So I'm imagining that's sort of an analytical exercise to do sort of like understanding of the patient population in terms of the biomarkers that are out there and sort of the diversity of them and which segments are growing and which segments are shrinking and how that relates to demographics, and then mapping that onto the way that you gather donor samples. Am I thinking about that right?
Yeah, you are. We have it's fairly sophisticated in terms of the thought process that goes behind it. I'll confess that in terms of how we manage it tool wise, we're doing something fairly simple in terms of, spreadsheets and, manually populated databases. But, overall, you're thinking about it exactly right. We're looking at what are kind of the donor populations that are available within these various databases, so that we can kind of forecast, okay, we want to go into this country, this country, this country. This is what the donor profile looks like. And we can then work with that and say, okay, well, you have XYZ kind of donors. We think we're going to need this. The other thing that we've done that's very interesting is we've started looking at donors that have more than one. We talk about HLA types that are more frequent in the population and those that are more rare. And so we've started to try and get populations where you get a donor that has more of those high frequency HLA types, so that we can get, when we get it into the manufacturing process, because these are cells, you don't always know what you're going to get. You go in and you think you're going to get one thing and the cells say, nope, you're going to get something else. And so we try and get more of those high frequency type of HLAs, so that when we go in and get the final product, we're more likely to have something that's going to be more usable for a broader part of the segment of the population.
Interesting. So I want to move over to the scale side. It sounds like you need to be able to, you're thinking about sort of different phases of bringing these manufacturing facilities up to scale. So you need sort of like the ability to manufacture small batches, but then also the ability to get up to commercial scale. So can you just walk us through how you think about that problem and how you, and what are the aspects that you look at each level to, understand your ability to scale?
Yeah. So in the earlier phases, it's really more about, how do I get in, get out, get my data, get out. So it's how fast can I get something into a CMO, get a manufacturing process. And for that, what we're looking for is, can we use standardized platforms, do we have what data do we already have that tells us this is going to work? Because we've had prior experience, whether it's with equipment or the process, but we want to be as efficient as possible when we get in. But we're not necessarily worried about how much product do we need, because we're not usually treating large populations in an early, if you're first in human trial, you're treating a handful of patients. So I'm not as worried about how many vials can I get, because often I can run, 6-8 lots and get more or less what I need for that first stage trial. But then when you look at, particularly if you're going into something like autoimmune, and I'm looking at, okay, now I'm in something where I'm faced with treating thousands of patients. Some of these autoimmune diseases start to get into the millions of patients. Obviously a small little benchtop, manually filling process, that's not going to cut it. So that's where you start to look at things like, okay, I need to start making investments into things like automation or significant scale. I want a closed system, because obviously that's going to give me more control, less contamination risk than an open process, more consistency. So, I think about things like, pipetting. We know that cells are very susceptible or they're very picky about how they're handled, if you will. They don't like to be mistreated. They don't like to be left out. And so your pipetting technique, when you're doing a manual process, can have a lot of impact on how happy your cells are. Do they grow? How much can you harvest in a particular lot? But when I'm looking at, and when I'm looking at a small scale, that might be fine, because I don't need that much product. But when I'm looking at a commercial scale, I might be starting to look at things like automation, because now I can say, okay, I need this mixing, I need XYZ speed, rotation, whatever it is. I can program that in, I can put it in a bioreactor. That gives me that consistency as well as the scale that I can then use. The trick is, where do you make that investment? Where do I make that pivot from, okay, I know I've got something that works, and now I've got to start building out this robust process that's going to serve my commercial population and be able to be validated and get through the regulatory process.
At each of those processes, I'm imagining that you need visibility into what's going right, what's going wrong, and where do you need to apply tighter degrees of control. So can you just walk us through from a data perspective? What are the lenses that you use at each of these scales to understand what's happening and to understand where you need to intervene or where you need to apply, a deeper degree of attention or control to improve the process?
One of the big things that we use is our continuous process monitoring program, which is run out of my manufacturing services group. So we track and trend the data. We understand what our critical and key parameters are. And we understand, particularly for the more developed programs, what are kind of the levers that we can pull that influence those parameters. And so the nice thing is by tracking and trending it, we can look at what are my set points, what are my targets, how am I doing, am I staying in control? And if we start to see a trend where things are dropping or we're constantly hitting out of limits, that gives us a signal that, okay, we have a problem here and we need to go in and potentially do an investigation, find out what's happening, and make some corrective actions.
Can you give us some examples of what are some of the metrics that you use that are in that quality control and management system?
Yeah, so we look at things like cell growth, cell viability, total viable cells. That tells us how the cells are doing, how's the lot expanding. We also look at things like number of vials that we harvested. What was the yield overall? What was the particulates within and the particulate reject rate? So those are all things that we trend on a regular basis. And we do this across both our commercial programs, but as well as our clinical. We like to start early and baseline that so that as we develop the program, we can tell, are we getting more efficient? Is the program, or excuse me, the product staying relatively the same throughout the development phases? So that when we get into commercial, we have a pretty good understanding of how this product is going to behave.
So are you using sort of the same assays, the same measurements at each of those phases so that you have like a one-to-one comparison? Or does the nature of your ability to measure at these different phases sort of change or alter like the comparisons that you would make between them?
A lot of the assays do stay the same. Things like cell growth, cell viability. Those are pretty standard assays, but some of them do change. You do get better on like your potency assays. How strong is your drug? How well does this work? That's probably the biggest one, and that gets refined throughout the development because you learn more about how does this behave. And so you can refine those assays as you go along, but generally they don't change drastically. But you are doing characterization work along the way that gives you more insight into that, and you might refine these assays as you go through the development process. But some of the basics in terms of how the cells behave, in terms of are they growing, are they producing cells, and how we're harvesting, those measurements all stay consistent throughout the entire development cycle.
How do you think about the data capabilities that you need to answer these questions and like developing them over time?
I think one of the things that we've done is we've done a pretty good job of being able to identify, what are those parameters that we need to be tracking and trending. I think for us, one of the things that I would like to be able to do more is have more automation in how we collect that data. Right now, it's very manual, and that's partly because we are using different CMOs, and they all have different processes, internal processes for how they collect and share that data with us, and we're somewhat dependent upon them to provide it to us. So that can make it a kind of labor-intensive process to go through because we're collecting things from like batch records, the analytical method results. A lot of that is often shared to us in kind of a paper form. So somebody going through and combing and pulling that data out can be very labor intensive to get the data that we need. So what I would like to see and what we've started to talk about is how can we automate that more so we can get more real-time access to that data? Because a lot of times if you can see it early, you can do an earlier intervention. You can say, okay, this looks like this lot is not doing well. Let's go back and look at the last 4 or 5 days. What did we do? And what corrections can we make to put this back on the right track? The other thing we do is we do tend to track how different donors perform. So it's not a one-for-one. You can get out of one donor collection, you can get multiple lots. We call them sister lots. And you can go back and say, okay, well, this lot looks like it's doing X, Y, Z. Let's go back and see, like the last time we used this donor, did we see a similar performance? That also tells us like, oh, this is kind of normal behavior for this particular donor, or last time it worked really well. It doesn't seem to be doing as well. So again, we need to go in and do kind of an intervention. And that data is also, tracked and trended through various applications.
So the CMOs that you're working with, they're literally mailing you paper, or it's like PDFs
No,
that they're sending.
No. GMP, we don't do that.
Yeah, right. So it's just in sort of like locked in these PDF type formats where the data's in tables that can't be read by a machine nor compared across the different CMOs that you're working with is how I'm thinking about that.
There's various tools that we use. We upload them to the shared data sites, but they're validated tools. And there's validated mechanisms that we use. And then we use what we call for information only, or FIO, where they can drop in kind of the unapproved data ahead of time. But it gives us a sense of, okay, this is what's happening, but as long as we all understand this is kind of your first cut, not the fully quality approved and reviewed version.
Right. So there's like the speed accuracy trade-off. And so you get the data as quickly as you can so that you can understand what's happening and, come up with some preliminary questions so that when the accurate data that arrives somewhat later, you can like determine whether those questions are noise or whether they're actually signals.
Exactly. And this is where things like electronic batch records can be helpful. And one of the things we always look at is, for a phase 1 product where I'm doing, again, those very small lots, is it worth the effort to go in and program for an electronic batch record system? Maybe not, because it's not that much data. It's fairly manageable. But then when you start looking into that more commercial robust, that's where we really like to do things like get into those electronic systems, because that allows for a little bit more, quicker turnaround of that data. You don't have to have somebody manually going in and pulling up a spreadsheet or whatever. They can download a report and we can get it a lot quicker.
Okay, so let's say that you had all of the data that's currently coming from your CMOs was in the format that you needed and sort of integrated together. Mm-hmm. Beyond that, what are the blind spots that you see on the horizon with regard to the manufacturing process that you think 1 year, a 3 year, a 5 year time horizon, you need to figure out how to get a handle on?
We tend to look at it very discreetly in terms of how is this lot doing ABC. We don't, and we do it from campaign to campaign, but what we aren't doing is tying it to taking it all the way through to the patient as an example. So I can look at my data right now and I can say, okay, from this campaign A to campaign B to campaign C, this is how we performed. The part that we haven't done, for example, is say, okay, this lot went into this patient and we saw this sort of response. And why? What made this maybe better or not better? Was it something about the patient? Was it something about that particular marker or product? So being able to get some of that, feedback loop of this particular donor worked well, this particular donor didn't seem to have as good of a response, and then how do we further refine the manufacturing process to be even more so we're having more efficacy when we come to treating our patients.
Yeah, that makes sense. And I would imagine that, there's all sorts of implications with trying to gather that data, that you're entering the world of sort of like regulatory requirements, of difficulty of interfacing with patients and getting the outcomes reported accurately from the provider, things like that. Are there any other challenges I'm missing?
Data privacy, the whole HIPAA, and then GDPR in Europe. So yeah, there are some significant challenges in getting, consent, getting consistent data from physicians. It's I think probably a little farther away than I would personally like, because I think that would make us, more effective. But I think it's something that as I'm, kind of looking at the industry, I think people are starting to think about more and more is how do we overcome those hurdles so that we can eventually get that data and make these personalized medicines even more effective.
There's a feedback loop between the therapeutic that you're delivering to the patient and then the outcome of the patient that allows you to then improve the therapeutic so that so that this patient and the next patient benefit from it.
And that's also data that you can use, if you're using a common platform like we are, that's also data that can then inform future products to make them even more effective as well and safe.
Right. So it feeds back into the research and development of the therapeutics themselves.
Exactly.
Interesting. I want to talk a little bit about on the manufacturing side. As you think about scaling up, I know from working with manufacturing organizations that cost is always a consideration, and that it's sort of like always top of mind. How can we produce obviously, the highest quality product, but as efficiently as we can, given that these processes can be, some of the most capital intensive processes that that biotech organizations engage in. So can you just walk me through, like, how do you think about reducing costs, becoming more efficient in the context of your manufacturing processes?
Yeah, so that's again where that process monitoring is really helpful. We're looking for things like waste and lot failure, and how do we address those to get more efficient and make those cost savings. And a good example, I mentioned that we trend our particulates. That's actually a really good story there, because we looked at it and we realized, oh goodness, we're losing a good amount of our product to particulate rejects. And so we discovered that by making a material change and doing some different things to our final filtration, we were able to reduce that reject rate by about 20%. And that translates into about a $2 to $3 million savings over just the first year that we've had that implemented. And then we've got another one that we've looked at in terms of if we make some equipment changes and put some investment into getting that change filed, that's another probably $4 million that we can save, on an annual basis in terms of waste. So I think those are two good examples of where you can, if you're looking at your trending data and kind of tracking back to what is happening here, why am I seeing what I am seeing, and then working back to what's an intervention that I can change, whether it's making a change to your process or change to equipment, becoming more efficient, you can realize some fairly significant savings.
It makes sense when you're looking at the trends that there's sort of like you can view it as like a predictive maintenance problem where, every so often you need to intervene with the machine, like clean it, something like that. But you can also view it as this component is not meeting our needs. We need to swap that component, or, whatever the people processes we have surrounding the machine are not sufficiently robust to or they're deteriorating. How do you sort of suss out how to approach the solution to a problem when you're seeing these trends in your metrics?
It's interesting. It's a lot of detective work, actually. You see the signal, okay, I think I have a problem. And then you have to go through this kind of methodical, almost, whether you call it a root cause analysis or 6M or whatever tool you're using, but you need to go through and kind of systematically look at it and say, what are all the things that could possibly be causing this? And then you get data to say, is this really what's causing it, or is this data that's not causing it? And why do you think that? And it's very interesting because, one of the things I see is that a lot of teams have bias, based on their previous experience. They want to jump to, well, of course it's this because it was this before. And so really pushing, the cross-functional teams, because usually we have our development people, we'll have manufacturing people, we'll have quality, we'll have QC, all together to say, no, you actually, yes, it could be that, but how do you know? What data do you have that actually demonstrates that is actually the problem? The other part of that is you have to be able to go in and talk to the people, because as you said before, there's the machine and there's the process itself, and then there's the people who surround that. And so being able to go in and observe, like, okay, Joe operator said, I'm doing XYZ, but then we went in and watched him and he's not actually doing XYZ, he's doing ABC, and that's part of my problem and I need to correct that. So it's a combination of looking at what data is available and then going out and kind of sense checking it with what do you actually see on the floor and does that match up? And that's often tells you where, do I have a data, do I have a process problem, or do I have a human problem that I need to go work through, training or better education or, coaching.
That makes a lot of sense. And I would imagine that, like, at some point in the detective process, if you sort of rule out that it's people and, there's something going on with the machine, there's sort of an experimentation process that needs to happen on the machine in order to determine whether, like, altering certain variables leads to the change in the outcome that you're talking about. But I also know that, running experiments on a machine is a cost, like you're taking that machine offline. So can you talk a little bit about how you think about sort of the value of, like, intervening with an experimental regime that helps you understand your process better versus the sort of the cost of taking machines offline and, like, making that kind of, and, like, getting the information you feel you need?
So first, these are validated systems, so we don't get to experiment on them. They have to run in their validated and compliant state. But what we do is we will do protocols and we'll run them in our development labs often, whether it's our own, or we contract out with the CMO and ask them to do that. But usually it's similar like for like equipment, and we try and do the similar conditions so that we can get as close to what we're seeing. And part of that depends on what your issue is. So if it's something that's more minor, where it's more of an improvement opportunity or a tweak to the process, we may make the decision to say, what, we're going to go ahead and continue to manufacture while we're investigating this and while the study is going on and we're collecting the data. If you're having, resolving an issue that's causing, quality and compliance issues in your final product that could harm product quality or patient safety, that's where you have to make the more difficult decision of, what, I'm going to take this down while I investigate what's happening. And I'm going to make the choice to stop until I know and I can implement a corrective action and then bring that back online. That's a more difficult, because now you're starting, even though we're manufacturing product ahead of time, particularly if you're in the middle of a run, that's not something that you take lightly, because it's usually takes time to bring it down. There's money, there's opportunity cost to that, and then bringing it back up is always a bit of a challenge.
Yeah, interesting. So it just sort of depends on, what you view as the downstream impact of the problem that's occurring, and sort of like how critical it is to like patients and to business operations, how broad sweeping of an intervention you're going to make in order to gather the information you need. So how do you think about risks in the manufacturing process? Things that might emerge in the future that you feel like you need to mitigate now, so that you don't have to do any of the interventions that we're talking about.
So we really do try and look ahead and look at what have historically been areas that have been problematic for us, and then what interventions can we do now, or investments can we do now to improve future performance. And a good example of this is, again, on one of our processes. It's very manual, it's open. We are looking at moving to an automated closed system, because that will get us more consistency. It's going to be an easier product to run, less contamination risk. And then that's an investment. So putting together the business case, how many lots have we, how many issues have we had based on contamination or operator issues? What do we think we're going to get? And then what's the investment? And then again, it's usually a 6-month to a 1-year process that you're looking at to get that actually implemented and ready to go. But knowing where your weak points are in your process, and then looking at things like technology, looking at your data, and being able to say, okay, if I do X, Y, Z, if I make this intervention now and make this investment now, I can get more product, I can get a better quality product, I will have less rejects. That's kind of the thought process that we go through.
Right. And so there's kind of a payback period on these kinds of investments, where you understand the timeline where the value accrues to you, or since those rejects are a known cost, you can sort of estimate in advance like when those costs are going to hit you, and so if you eliminate them, that gives you your ROI. I'm imagining.
Yes, that's exactly right.
Awesome. What systems do you need to have in place in order to understand your manufacturing process better, in order to be able to, accomplish the goals that the organization needs to accomplish? What does that look like for you?
I think that one of the biggest challenges that we have is, again, a lot of the data that we have available to us. We have smaller orphan indications, smaller lots. So it's very challenging for us. I would like to get, more data collected, because for me, it's hard sometimes to tell what is actually a signal that's I've got, something that I need to be working on, or what's just noise in the system, because you're working with such small data sets. So I think one is, how do I get access to more data in general? Because we're working with, something that's got, 6, 8, 10, 12 lots. You can't get statistically significant, and meaningful off of that. But I think, so if there's a way that we can either leverage what other people have, if there was something, commercially available, or just getting more experience in patients and bringing that data in so that we can look at it and understand, okay, this is really something that we need to address. I think for us, the other thing is better tools. I think because we're small, we've chosen to not make a lot of the investment and do a lot of this manually. And I've actually been chewing my IT, VP's ear a little bit on this. I'm like, this would really help me if I had some more automation to be able to do it. And he's like, yes, we know, but this is where you make the choices of where you want to invest your money. And I think if we can get some of those tools in, and then one example is, we talked about those electronic batch records. There's some modules that you can get that will actually then, if you implement them, will do some of that automated tracking and trending. Instead of doing like kind of a data dump, it will actually do some of that work for you. And it's an investment that we would need to make, and we would need to probably partner with our CMOs that have those batch records. But having that ability to have that again, just kind of at our fingertips, would be hugely useful for us.
Yeah, interesting. So there's also what's occurring to me as I'm hearing you talk is just like at the scale you're at with the number of patients that you're engaged with, you want as much data as you can to understand what that manufacturing process is going to look like and what sort of problems you're going to encounter, but also you're sort of constrained by the scale and like can only draw the conclusions that the data enables you to.
That's right. And one of the things that, I'm always kind of worried about is data overload. There's also the concern that, just because I have more data doesn't necessarily mean it's going to give me a better outcome. It just may be more data that I then have to sort through and figure out, does this really mean something for me or not? So we got to be, very thoughtful about what do I really need and not just pull everything in just because I have access to it.
Yeah. Do you have a list of like, what are the 3 key questions that you need that you need answers to that you would be excited to be able to answer, once the data is available?
One thing I would love to know, and we've talked about this, is how do I know when I put a donor through, is what's my guarantee that this is going to be, ultimately result in a great product? Is there a marker? Is there some sort of, biomarker or something that I could look for, we could screen for early on that's going to tell me, yes, this is going to get you a high yielding, high performing lot every time? I would love that. That would be phenomenal.
So it's like a predictive QC measure,
Exactly.
like a machine learning model that looks at like all of the data that's coming through and gives you an indication of, this is the expectation of, your quality assay metrics, or this is the probability that we think that this is a high quality product.
Yeah, that's probably questions 1, 2, and 3 for me.
Interesting. Okay. Well, just as we draw toward a close, I'm just interested in sort of zooming out with you and like how you think about biotech manufacturing more broadly. I'd be interested in hearing from you, like, what are the technologies you're most excited about applying to the manufacturing process in the years to come, or that you see as like sort of emerging that and might have, a great impact on the work that you do?
It's interesting. One of the things that it's kind of a buzzword right now, but we talk about AI a lot. And I'm very interested in where is that going to take the industry? And again, it comes back to how do you build those data models so you can get more predictive? I think that this is something that we're currently, I don't want to say sitting out, but we're kind of taking a wait and see. But I think as that whole part of the data industry expands, I think there's going to be opportunities to really get some good modeling and some good tools available. And that can really help us, because if I can do things where I can do that predictive modeling ahead of time, and I can save, running manufacturing that's not ultimately going to be successful, I think that's a huge win for the company and for patients, because then we're not spending resources on things that ultimately aren't going to work for them. I think the other thing, particularly in cell therapy, is it's such a new industry, and we really haven't settled on what's kind of the standard platform that we're going to use. So I think as we develop this technology and the equipment and what's going to be kind of our standard basis for manufacturing, and how do we take it from that bench scale to that large commercial scale, I think that will be, for me, particularly helpful. We're even seeing when we go out and look at, everybody has their own platform, they have their own set of equipment. So every time you put in a new process, it's another investment into how do I do this? And having somebody that says, and we've run this, we've modeled this, we know this works, here's the data that supports that. And you can look at it and you're like, okay, I know if I put my money here, this is going to be something that's going to eventually get me a robust commercial program that I can run.
Well, Amy, it's been a pleasure to have you on the podcast. I really appreciate the time, and I'll look forward to connecting down the line.
Sounds great. Thank you very much for having me.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.